Good morning teachers, seniors and batchmates. Today I am presenting seminar on structural chromosomal abnormalities. I will be dealing under the headings of what are chromosomes, structure of chromosomes, history of chromosomes, structural chromosomal abnormalities, types of structural chromosomal abnormalities, causes of structural chromosomal abnormalities, diagnosis of structural chromosomal abnormalities, uses or applications of studying cytogenetics or structural chromosomal abnormalities. Chromosomes are bundles of tightly coiled DNA located within the nucleus of almost every cell in our body. Each chromosome contains thousands of genes which make proteins that direct the body's development, growth and chemical reactions. Each human cell normally has 23 pairs of chromosomes, that is 46 chromosomes. Half of the chromosomes come from mother and half come from the father. The first 22 pairs are called autosomes. The 23rd pair is called X and Y chromosomes. They consist of sex chromosomes. Females usually have two X chromosomes, one from the father and one from the mother, and males usually have one X and one Y chromosome in each cell, coming one from father and one from mother. This is a photomicrograph to show 23 pairs of chromosomes in our body. First chromosome being the largest, and the last pair of chromosomes are called as sex chromosomes, which are comprised of X and Y chromosome. First 22 pairs, that is from chromosome number 1 to 22, are same in males and females. The last pair of chromosomes differ in males and females. Females have two X chromosome coming one from father and one from mother, whereas males have one X and one Y chromosome coming from each parent. According to International System of Human Cytogenetics Nomenclature, centromere divides the chromosome into short arm P and long arm Q. The region adjacent to centromere of short arm and long arm are given numbers as P1 and Q1 respectively. The next distal region is given as 2 and so on. The regions are subdivided into bands and bands are subdivided into subbands as the resolution increases and the numbering done sequentially. As we can see in this photomicrograph, this is a chromosome, this is a centromere, this is a short arm of the chromosome and this is a long arm of the chromosome. From the centromere, it is divided into region 1, region 2, region 3. Similarly, long arm divided into region 1 up to here, region 2 till here, and this is region 3. Each region is further divided into bands as region 1, band 1, region 1, band 1, region 1, band 2, region 1, band 3. Similarly, in the long arm, region 1, band 2, region 1, band 3, region 1, band 4, region 2, band 1, region 2, band 2, region 3, band 1, region 3, band 2, region 3, band 3. And these bands are further subdivided into subbands as we can see here. Region 1, band 1, subband 1, region 1, band 1, subband 2. In the long arm, region 1, band 2, subband 2, region 1, band 4, subband 1, region 1, band 4, subband 2, region 1, band 4, subband 3, so on so forth. As the resolution increases, the division goes on increasing. History of chromosomes. Chromosomes were first described by Strasberg in 1875. Arnold first observed chromosome in 1879. The term chromosome, however, was first coined by Wallier in 1888. Hansenmann and Fleming counted the chromosome in 1891. The word chromosome comes from Greek word chromo means color and soma means body, describing their strong staining by particular dyes. Winnewater isolated X chromosome, Painter isolated Y chromosome, Tigio and Levin described correct number of chromosomes, that is 22 pairs of autosomes and 2 sex chromosomes in 1956. Classification of chromosomes based on position of centromere. Chromosomes are classified by their length and the position of their centromeres. The centromere is the point at which the two identical strands of chromosomal DNA, the sister chromatids, attach to each other during mitosis. The location of the centromere is used to classify chromosomes as metacentric, submetacentric, acrocentric, and telocentric. In metacentric, centromere is exactly in the middle, and chromosomes number 1, 3, 19, and 20 come under this category. In submetacentric, centromere is located slightly away from the center, such that one arm is short and other arm is long. That is, it divides the chromosome into short P arm and long Q arm. Chromosome numbers 2, 4, 12, 16, 18 and X chromosome come under this category. Acrocentric chromosomes. Here centromere is located very near the end such that one arm is very short and other arm is very long. Chromosomes number 13, 14, 15, 21, 22 and Y chromosome come under this category. Telocentric chromosomes. Here centromere is located at one end such that only long arms are present. This is a photomicrograph to show different type of chromosomes classified based on centromeres. Here in metacentric chromosome, the centromere is exactly in the center, whereas in submetacentric, the centromere is slightly away from the center, producing one long arm and one short arm. In acrocentric chromosome, the centromere is very much near the end, leading to production of one very short arm and one very long arm. Whereas in telocentric chromosome, centromere present at the tip, leading to production of only long arms. There are no short arms. Chromosomal abnormalities. In 1959, two discoveries opened a new era of genetics. Jerome Lejeune, Marthe Gautier and M. Raymond Turpin discovered the presence of an extra chromosome in Down syndrome patients. C. E. Ford and his colleagues P. A. Jacobs and J. A. Strong first observed sex chromosomes anomalies in patients with sexual disorders. Most chromosomal abnormalities occur as an accident in the ovum or sperm. They may occur prenatally, postnatal or pre-implantation periods. Abnormalities happening after conception can result in mosaicism, where some of the cells have normal chromosome number and others have different number of chromosomes. Chromosomal abnormality can be inherited from a parent or be de novo. There are various types of chromosomal abnormalities which can be broadly classified into two groups, that is numerical and structural. In numerical, there is defect in the chromosome numbers, that is there may be either more than 23 chromosomes or there may be less than 23 chromosomes. Whereas in structural abnormality, the number of chromosomes remains same in a cell, but there is alteration in structure of chromosomes leading to loss, rearrangement or gain of some chromosomal segments. Structural chromosomal abnormalities lead to changes or alteration in the normal structure of chromosome. They alter the number of genes, their sequence or kind of genes present in the chromosomes, and do not alter or change the number of chromosomes. These changes may either take place in a single chromosome or more than one chromosome. On this basis, it is of two types, that is intrachromosomal abnormalities or interchromosomal abnormalities. In intrachromosomal abnormalities, abnormalities remain confined to a single chromosome of a homologous pair. In interchromosomal abnormalities, in this, breaks occur in non-homologous chromosomes and the resulting fragments are interchanged between the two non-homologous chromosomes. Common structural abnormalities seen are deletion, inversion, duplication, translocation, ring chromosome, isochromosome, dicentric chromosome. Deletion. Deletion refers to loss of portion of segment of a chromosome resulting in loss of a single gene or part of gene. Depending upon the location, deletions are of two types. One is terminal deletion, other is interstitial deletion. Terminal deletion. Here breaks occur near the end of chromosome. In terminal deletion results from a single break in a chromosome arm producing a fragment with no centromere which is then lost at the next cell division and the chromosome bearing a deletion is formed as shown in this photomicrograph. This is a chromosome with segment A, B, C, D, E and F. Break takes place here and the broken segment A gets lost as it is shown here, and this segment ends are protected by acquiring telomeric sequence. Interstitial deletion occurs when there are two breaks within the chromosome arm followed by loss of chromosomal material between the breaks and fusion of the broken ends. One can specify in which region and what bands the breaks have occurred. Interstitial deletion. Here breaks occur at 2 points and the broken ends of the origin chromosome get fused to reunite, resulting in loss of a fragment of chromosome, as seen in this photomicrograph. Here we can see this is a chromosome. Breaks are happening at 2 points, at point here and at point here, and this segment, CD segments, gets separated and it gets lost in successive cell division. These two broken ends fuse, forming chromosome with the deleted part or the lost part. DNC part is lost. This is another photomicrograph to show interstitial deletion, where breaks are happening at 2 points here and here in the long arm, and this part gets lost. Inversion. Inversion refers to a rearrangement that involves two breaks within a single chromosome with reincorporation of the inverted intervening segment, resulting in a chromosome in which a segment is oriented in a reverse order. The inverted segment is rotated to a full 180 degree, leading to a change in arrangement of gene loci. This is a photomicrograph to show inversion. This is a chromosome. The break happens at one point here and other point here, and this segment rotates 180 degree and gets inserted upside down. That is, this end comes up and this end comes down. Depending upon whether centromere is involved or not, inversions are of two types: paracentric and pericentric. In paracentric inversion, an inversion occurs in one arm of the chromosome. That is, centromere is not involved. Both breaks occur in one arm of the chromosome. In pericentric inversion, centromere is involved. That is, breaks occur on each side of the two arms of a chromosome. Centromere is included in the detached segment. That is, breaks are seen on opposite sides of the centromere. Inversions are often fully compatible with normal development. This is a photomicrograph to show two types of inversions, that is, paracentric and pericentric. In paracentric, breaks happen on only one arm of the chromosome. This is one chromosome and this is a centromere. Breaks happening on only in the long arm. We can see in this figure, that is at point B and point D. This segment BCD rotates 180 degree and gets inserted in reverse form as DCB. Whereas in pericentric, there is division happening on either side of the centromere. This is centromere and there is a break point happening at segment D and segment F. And this DEF segments rotates 180 degree and gets inserted in reverse form as FED. That is, the centromere is involved in the break. This is another photomicrograph to show pericentric and paracentric inversions. In paracentric, the divisions happen only on one side of the centromere, that is at here and here, and this gets rotated by 180 degree and gets inserted in a reverse form. Whereas in pericentric, there is break happening on either side of the centromere. On either side of centromere, one break is here, other break is here, and it gets rotated by 180 degree and gets inserted in a reverse direction as shown here. The top has come to down and the down part has gone to up. Effects of chromosomal inversion. It helps in retaining the original gene combination by reducing the crossing over frequency. Inversion enhances the position effect and thereby produces phenotypic changes. Inversion can cause chromosome polymorphism within the population, leading to karyotype evolution. Inversions produce balanced gene complexes, that is supergenes, through reduction of crossing over, which may in turn confer greater biological fitness and adaptability. Insertion. Here a portion of one chromosome is deleted and gets inserted into another chromosome, as seen in this photomicrograph. This is one chromosome, this is another chromosome. This chromosome undergoes break at one point here and one point here, and this broken segment or deleted segment gets inserted into another chromosome at point here, leading to formation of this chromosome. So this is a chromosome with a deleted segment and this is a chromosome with gained segment. Duplication. Here a portion of chromosome is duplicated, resulting in extra genetic material. As shown in this photomicrograph, this is a chromosome. This segment from here to here undergoes duplication and gets inserted side by side in the same sequence. That is, this is one gene, same sequence, side by side. Duplications. Duplications result from doubling of chromosomal segments and occur in a range of sizes and locations. Types of duplication. There are three types. 1. Tandem duplication. 2. Reverse tandem duplication. 3. Displaced duplication. In tandem duplication, the extra segment or duplicated segment and the parent segment are next to each other and have the same order of genes. Whereas in reverse tandem duplication, the extra segment lies next to parent segment with reversed gene sequence. In displaced duplication, the duplicate segment lies some distance away from the parent segment. This is a photomicrograph to show different types of duplications. This is a chromosome with segment A, B, C, D, E, F, G, H, I. Segment B and C undergoes duplication in tandem duplication. The duplicated segment lies next to parent segment in the same order. That is, B and C is a parent segment and the duplicated segment B and C lies next to parent segment. In reverse tandem duplication, the B and C segment undergoes duplication but gets inserted next to it in a reverse form as C and B. In displaced tandem duplication, B and C segment undergoes duplication. The duplicated part gets inserted away from parent segment. Effects of chromosomal duplication. Duplications are more frequent and less deleterious than deletions. Duplications are believed to be the most important raw material of organic evolution. Large duplications affect the regulation of gene activity and thereby causes phenotypic alterations, which in turn promotes speciation and evolution. Translocation. Translocation is a process in which part or segment of chromosome cross over and exchange genetic material. There are two types of translocation, that is intrachromosomal translocation and interchromosomal translocation. In intrachromosomal translocation, change in position of a segment within the chromosome either from one arm to other or from one location to another in the same arm. So based on this, again there are two types. Intraradial, where translocations from one location to another in the same arm takes place. In extraradial, translocation from one arm to another arm takes place. In interchromosomal translocation, transfer of a segment from one chromosome to other takes place. In non-reciprocal type of translocation, there is only one-way transfer of a chromosome segment from one chromosome to another chromosome. In reciprocal translocation, there is mutual exchange of segment between two chromosomes, that is, exchange of chromosomal segments between different non-homologous chromosomes takes place. This is a photomicrograph to show different types of translocations. This is here the non-reciprocal intrachromosomal translocation, where the segment B and C undergoes translocation to this arm between F and G here, as seen in this chromosome. This has got translocated to here, leading to formation of this chromosome. In non-reciprocal interchromosomal translocation, the segment B and C gets translocated to another chromosome between N and O here, leading to formation of this chromosome. That is, this is a chromosome which has lost segment B and C from here, and this is a chromosome which has gained the segment BC at this region. In reciprocal interchromosomal translocation, the segments get interchanged between two chromosomes, between these two. This is one chromosome, this is another chromosome. The segment AB from this chromosome gets translocated to here, and this MNO segment of this chromosome gets translocated to this region, as seen here. Reciprocal translocation. Reciprocal translocations are balanced if there is no net loss of genetic material. Each chromosomal segment is translocated in its entirety. If this is present in gametes, that is sperm or ova, all somatic cells in progeny keep the abnormal chromosomal structure. Balanced translocations are not usually associated with loss of genes or disruption of vital gene loci, so most carriers of balanced translocations are phenotypically normal. While most patients with balanced translocations are asymptomatic, 6% have an associated disorder such as autism, decreased intellectual ability, and congenital abnormalities. In such instances, it is likely that the abnormalities are the result of disruption of a gene located at the breakpoint of the translocation. Robertsonian translocations. Robertsonian translocation involves the centromere of acrocentric chromosomes, that is, it occurs in chromosome 13, 14, 15 and 21. When two non-homologous chromosomes are broken near the centromere, they may exchange two arms to form one large metacentric chromosome and a small chromosomal fragment. The latter lacks a centromere and is usually lost in subsequent divisions. As in reciprocal translocation, Robertsonian translocation is balanced if there is no significant loss of genetic material. This is a photomicrograph to show Robertsonian translocation. Here, break happens near the centromere, and these two segments get fused and these two get fused, as we can see here. There are two short arms in this chromosome and there are two long arms in this chromosome. Effects of Robertsonian translocation. If a carrier is fertile, however, his or her gametes may produce unbalanced translocations, in which case offspring may have congenital malformations. Robertsonian translocations of chromosome 13 and 14 are most common and are seen in 0.97% of 1,000 newborns. A Robertsonian translocation of chromosome number 21 imparts a greater risk of having a child with Down syndrome. Maternal transmission is more common than paternal. Ring chromosome. Ring chromosome is a chromosome whose arms have fused together to form a ring. Ring chromosomes are formed by a break involving both telomeric ends of a chromosome, deletion of the eccentric fragments, and end-to-end fusion of the remaining centric portion of the chromosome. The consequences depend primarily on the amount of genetic material lost because of the break. The abnormally shaped chromosome may impair normal meiotic division, but in most instances, this chromosomal abnormality is of no consequence. This is a photomicrograph to show ring chromosome formation. That is, this is one chromosome, breaks happening on either side of the centromere. That is, one break is here, other break is here. So these two parts gets lost and these two raw ends of the chromosome get fused as shown here. This is another photomicrograph to show ring chromosome formation. That is, this is a chromosome, this is long arm of chromosome, this is short arm of chromosome, this is the centromere. There is a break happening at the telomeric end. That is, one break is here and other break is here, and these two broken ends, they get lost in successive cell division, whereas these two raw ends fuse to form a ring chromosome. Effects of ring chromosomes. Ring chromosomes have been associated with epilepsy, mental retardation and dysmorphic faces, dwarfism and microcephaly, as well as Turner syndrome. Ring chromosomes also may be found in a variety of human malignancies, soft tissue tumors of adipose tissue, and in Fanconi's anemia. Isochromosomes results when the centromere divides in a transverse plane rather than in a normal long axis of a chromosome, leading to formation of one with two short arms and one chromosome with two long arms. As seen in this photomicrograph, normal division of chromosomes takes place in longitudinal direction like this during mitosis, whereas here the division happens transversely in this direction, leading to formation of one chromosome with two short arms and another chromosome with two long arms. Dicentric chromosome. It is formed through the fusion of two chromosome segments, each with a centromere, resulting in the loss of acentric fragments lacking a centromere and the formation of dicentric fragments. Their existence has been linked to certain natural phenomena such as irradiation and have been documented to underlie certain clinical syndromes, notably Kabuki syndrome. This is a photomicrograph to show dicentric chromosomes. There are two chromosomes, this is one and this is one. There is a break happening at one end of the chromosome here and one end of the chromosome of this chromosome here. These two broken ends fuse and these two fragments without a centromere get fused as shown here. The two long segments have fused, forming one chromosome with two centromeres, whereas these two tiny fragments have fused without a centromere. So these gets lost in successive cell divisions. Coming to chromosomal nomenclature. Different types of chromosomal abnormalities have been given different symbols or alphabets or name or some number. As we can see here, numerical designation of autosomes have been given number as chromosome 1 to 22, whereas sex chromosomes have been given name as X and Y. Addition of a whole or part of a chromosome is indicated by symbol plus, whereas loss of a whole or part of a chromosome is indicated by symbol minus. Numerical mosaicism is indicated by the slash, where some of the cells are having 46 number of chromosomes and some of the cells having 47 number of chromosomes. Short arm of chromosome is designated as small letter p. Long arm of chromosome is designated as small letter q. Isochromosome are indicated by capital letter I. Ring chromosome are indicated by capital letter R. Deletions are indicated as DEL. Insertions as INS and translocations as T. Derivative chromosome as DER. Terminal chromosome as TER. Chromosomal instability syndromes. These are group of disorders characterized by chromosomal instability and breakage. They often lead to an increased tendency to develop certain types of malignancies. Coming to causes of structural chromosomal abnormalities. These structural chromosomal abnormalities can happen accidentally or what we call as idiopathic, or they may happen due to exposure to radiations, toxic chemicals, some type of viral or bacterial infections, medicines, certain drugs, alcohol or tobacco. Consequences of structural chromosomal abnormalities. Chromosomal abnormalities can have significant clinical consequences leading to spontaneous abortion, stillbirths, neonatal deaths, hospitalization, congenital malformations, intellectual disability, and an identifiable syndrome. Diagnosing chromosomal abnormalities by various techniques. One is karyotyping, in situ hybridization, fluorescent in situ hybridization, spectral karyotyping or multicolor fluorescent in situ hybridization, and comparative genomic hybridization. Karyotyping. Karyotype refers to the entire set of chromosomes in an organism. Karyotyping is the procedure of obtaining structure of chromosome. For karyotyping, rapidly dividing cells are taken. Rapid dividing cells taken for karyotyping are the peripheral blood lymphocytes, skin fibroblasts, bone marrow cells, amniotic fluid cells, or chorionic villi. Procedure of karyotyping. Here, cells are inoculated and cultured for 10 to 11 days at 37 degrees. Colchicine is added to arrest cells in metaphase. After incubation and centrifugation, the medium is replaced with a hypoosmotic solution to produce cell lysis. And the leftover chromosomes are fixed on a glass slide, stained with a specific dye, observed under light microscope. This is a photomicrograph to show different phases of cell division in mitosis. And for karyotyping, the cells have to be fixed under this stage, that is metaphase, to be observed under microscope. This is a photomicrograph in short to show the entire process of karyotyping, where WBCs are obtained from peripheral blood are cultured. WBCs are stimulated to divide and then arrested in metaphase with chemicals like colchicine. Then hypotonic solutions are used to release chromosomes from the cell. And the released chromosomes are fixed and stained by one of the several techniques and are observed under microscope. Fluorescent in situ hybridization. This method uses fluorescent labeled DNA probes to assess the DNA sequence of interest in an individual's genome by hybridization. In addition to metaphase, fish cells can be analyzed at a variety of stages like interphase, which increases the number of cells available for examination. This facilitates study of cells which do not progress to metaphase Example, amniotic cells, where interface analysis is done. One specialized type of interface analysis involves the application of fluorescent in situ hybridization to paraffin embedded sections, thereby preserving the architecture of the tissue. In addition to standard metaphase and interface fluorescent in situ hybridization analysis, a number of enhanced techniques have been developed for specific types of analysis like multicolor fish techniques, reverse painting, fiber fluorescent in situ hybridization, comparative genomic hybridization, spectral karyotyping, and multicolor fluorescent in situ hybridization techniques. Applications of cytogenetics or study of structural chromosomal abnormalities. Study of cytogenetics helps in diagnosis and classification of leukemias, diagnosis and confirmation of various syndromes, prediction of prognosis and outcome of diseases, disease staging, selection of patients for targeted therapy, monitoring of minimal residual disease, prediction of drug efficacy and toxicity, aids in studying evolutionary changes. Applications of fluorescent in situ hybridization. The use of interface fluorescent in situ hybridization for analysis of amniocentesis samples can be performed rapidly, that is within 24 to 48 hours, to detect chromosomal abnormality prenatally. Another area in which interface analysis is routinely utilized is cancer cytogenetics. Many site-specific deletions, duplications, translocations are associated with specific types of malignancies. This is a photomicrograph to show multicolor fish, where each pair of chromosome is given different fluorescent colors, and different type of chromosomalities can be seen in various chromosomes simultaneously at one point. In this photomicrograph, we can see translocation between chromosome pair 6 and chromosome pair 16. That is, a part of or segment of chromosome is translocated from here to here and from here to here. Also, we can see translocation between chromosome pair 2 and chromosome 16. We can see a piece of this segment of this chromosome is translocated here, and this piece has come to here. Also, there is inversion seen in other part of this translocated chromosome. Now let us see various chromosomal abnormalities seen in various clinical syndromes diagnosed through cytogenetics. Coming first one to Prader-Willi syndrome. Here the structural chromosomal abnormality is deletion of long arm of chromosome 15 at region 1 band 1 to region 1 band 3. Individuals with this type of deletions presents with obesity, muscular hypotonia, short stature, hypogonadism, narrow bifrontal diameter, almond-shaped eyes, and strabismus, as seen in these photomicrographs. Angelman syndrome. Here deletion is same as Prader-Willi syndrome. That is, there is deletion on the long arm of chromosome 15 at region 1 band 1 to region 1 band 3, similar to Prader-Willi syndrome. Here individuals affected present with abnormal puppet-like gait and paroxysms of laughter due to brainstem defect not associated with happiness. Therefore, this syndrome is also known as happy puppet syndrome. Above two macrodeletion syndromes, that is Prader-Willi syndrome and Angelman syndrome, have similar chromosomal abnormality. Insight into the pathogenesis revealed imprint disorder, that is, parent of origin of the deletion determines the type of anomaly. If the deletion is paternal, the result is Prader-Willi syndrome, whereas if the deletion is maternal, the result is Angelman syndrome. Now, Cri-du-Chat syndrome. Here, the structural chromosomal abnormalities deletion of short arm of chromosome 5 at region 1 band 5. Individuals affected with this syndrome presents with microcephaly, round face, hypertelorism, epicanthal folds, downward slanting of palpebral fissure, strabismus often divergent, low set poorly formed ears, facial asymmetry, and a typical cat-like cry, mewing of a cat due to abnormal laryngeal development, which becomes less pronounced as age increases. Wolff-Hirschhorn syndrome. Here the chromosomal abnormality is deletion of short arm of chromosome 4 at region 1 band 6 and subband 3. Individuals affected with this abnormality present with broad flat nasal bridge, widely spaced eyes and high forehead, giving Greek warrior helmet appearance. Short distance between nose and upper lip, downturned mouth with delayed growth, developmental intellectual disability and seizures. Cat eye syndrome. Here the chromosomal abnormality is inversion and duplication of in long arm of chromosome 22 at region 1 band 1. Individuals affected with this abnormality present with colobome of the iris, which appears as a cleft or gap in the iris, and the elongated pupil gives the appearance of a cat's eye, as seen in the photomicrograph. We can see in the pupil looks like slit or cleft, giving a cat's eye appearance. Individual with this defect can also present with anal atresia and fistula. Coming to Kibuki syndrome. This anomaly is associated with dicentric chromosomes. It is named so because of the facial resemblance to a stage makeup used in kabuki, a Japanese theatrical form. Individuals present with long eyelashes, depressed nasal tip, ear deformity, hypotonia, ptosis, short stature also can be associated with gastrointestinal and cardiac defects. Neurofibromatosis 1. Here the defect is deletion in long arm of chromosome 17 at region 1, band 1 and subband 2. Individuals with this defect presents with cutaneous and subcutaneous neurofibromas. These tumors may be more than 500 and appear as soft pedunculated masses, usually about 1 cm in diameter, as shown in the photomicrograph. We can see several pedunculated masses. Occasionally, they may reach alarm proportion up to 25 cm and dominate the physical appearance of a patient. Coming to Duchenne muscular dystrophy. Here the defect is deletion in the short arm of chromosome X at region 2, band 1 and subband 2. Individual affected with deletion present with progressive proximal muscular dystrophy as shown in this photomicrograph. As the child goes on growing, the muscular dystrophy goes on increasing, and by the age of 15 the child is wheelchair bound, and by the age 20 the individual is dead. And there is associated increased serum creatine kinase. There is myofiber degeneration with fibrosis and fat infiltration in the muscle. Some other chromosomes associated with chromosomal abnormality. WAGR syndrome, where the defect is deletion in the short arm of chromosome 11 at region 1 band 3, and such individuals present with Wilms tumor, aniridia, genitourinary abnormalities like undescended testis, hypospadiasis in males, and internal genitalia and urinary abnormalities in females, also associated with mental retardation. Beckwith-Widman 1 syndrome, where the defect is duplication in the short arm of chromosome 11 at band 1 at region 1 band 5, and individuals present with exomphalos, macroglossia, gigantism in the neonate. Charcot-Marie Tooth type 1A syndrome, where the chromosomal defect is duplication in the short arm of chromosome 17 at region 1 band 2 and subband 2. Individuals with this defect present with progressive muscle wasting, weakness and nerve conduction defects. Now Charge syndrome. The chromosomal abnormality is deletion of long arm of chromosome 8 at region 1 band 2 subband 2, and the individuals with this defect present with colobome of eye, heart anomaly, atresia of Koina, mental retardation and somatic developmental delay with ear abnormalities like deafness. Retinoblastoma. The defect is deletion in the long arm of chromosome 13 at band 1, region 1 band 4 and subband 1-1, and the defect is retinoblastoma, that is tumor of the eye. Glyceryl kinase deficiency, and the defect is deletion in the short arm of chromosome X at region 2 band 1 and subband 2. Individuals with this defect present with mental retardation, growth retardation, elevated urinary glycerol and pseudo hypertriglyceridemia. Kallmann 1 syndrome. The defect is deletion in short arm of chromosome X at region 2, band 2 and subband 3-1. Individuals with this defect present with congenital idiopathic hypogonadotrophic hypogonadism and anosmia. Miller-Dicker or Leeson-Cephali, where the defect is deletion in the short arm of chromosome 17 at region 1, band 3 and subband 3. With this defect, individuals present with Leeson-Cephali, that is partial or complete absence of gyration of the cerebrum, macrocephaly, prominent occiput, anterior narrow forehead, cardiac malformations, hypoplastic male external genitalia and mental retardation. DGeorge-1 syndrome, where the defect is deletion in the long arm of chromosome 22 at region 1, band 1, subband 2 and 1. Individuals with this defect present with hypocalcemia due to hypoplasia of parathyroid, thymic hypoplasia, defect in outflow of heart. DGeorge-2 syndrome, where the defect is deletion in the short arm of chromosome 10 in region 1, band 4. Here, the clinical features are similar as DGeorge-1 syndrome, but there is associated mental retardation and dysmorphic features of face and skeletal system. Now coming to application of fluorescent in situ hybridization in acquired chromosomal abnormalities seen in different type of malignancies or neoplasms. In addition to the constitutional cytogenetic chromosomal abnormalities that are present at birth, somatic chromosomal changes can be acquired later in life and are often associated with malignant conditions. Somatic changes can be deletion, duplication or translocation, as we just now studied. Chronic myeloid leukemia. Here, the chromosomal abnormality is translocation between chromosome 9, that is long arm of chromosome 9 at region 3 band 4 to translocation to chromosome 22 at region 1 band 1. These regions are known as BCR-ABL region as shown in the photomicrograph. This is chromosome number 9, this is chromosome number 22. This part of the chromosome known as ABL chromosome gets deleted and gets translocated here in this region, leading to formation of this chromosome. This translocation is known as BCR-ABL translocation, and this chromosome is known as Philadelphia chromosome. It is said so because this was first identified or detected in a place called Philadelphia. This type of abnormality or this Philadelphia chromosome is seen in about 92% of chronic myeloid leukemia patients. Other abnormalities seen in chronic myeloid leukemia patients are deletion in the long arm of chromosome 9, duplication in chromosome 8, inversion in long arm of chromosome 17. Additional abnormalities or mutations seen in blast crisis of chronic myeloid leukemia patients are frequent mutations seen are duplication of Philadelphia chromosome, isochromosome formation of chromosome 17, loss of 5 chromosome. Rare mutations seen are translocation between chromosome 15 and chromosome 7 and translocation between chromosome 3 and inversion in chromosome 3. Acute myeloid leukemia, which are broadly classified as being favorable, intermediate and poor prognostic types, extensive numerical or structural chromosomal abnormality and myelodysplastic background type of acute myeloid leukemias are very aggressive and have poor prognosis. Whereas acute myeloid leukemia due to translocation between chromosome 15 to 17 or chromosome between 8 and 21 and inversions in chromosome 16 or translocation between chromosome 16 to 16 have shown favorable prognosis. Coming to lymphoproliferative disorders. One among them is chronic lymphocytic leukemia. Only 50% of chronic lymphocytic leukemia patients have detectable chromosomal abnormalities. Commonest structural abnormalities seen is deletion in long arm of chromosome 13 and deletion in long arm of chromosome 14, chromosome 17 and long arm of chromosome 6. In multiple myeloma, commonest structural abnormalities seen are translocation between chromosome 14 to 16 and 4 to 14. Acute lymphoblastic leukemia. Most commonest chromosomal abnormality noted is translocation between chromosome 9 and 22. Chromosomal abnormalities with favorable prognosis are hyperliploidy, that is chromosome numbers exceeding more than 52, and translocations between chromosome 12 and 22 have shown favorable prognosis. Whereas hyperploidy, where there is chromosome number less than 40, and translocations involving between chromosome 1 to 19, 9 to 22, 4 to 11 and 11 to 19 have shown poor prognosis or unfavorable prognosis. Non-Hodgkin's lymphoma. In follicular lymphoma, translocation between chromosome 14 and chromosome 18 is seen. In Burkitt's lymphoma, there is translocation between 8 to 14 and between 2 and chromosome number 8. In mantle cell lymphoma, there is translocation between chromosome 11 and 14. In diffuse large B-cell lymphomas, there is translocation between chromosome 3, 22 or translocation between chromosome 3 and 14. In anaplastic large cell lymphomas, there is translocation between chromosome 2 and 5 and between 1 and 2. Polycythemia vera. Most common abnormalities seen in polycythemia vera are duplication seen in chromosome number 8, deletion seen in long arm of chromosome 7 and deletion in long arm of chromosome 11, deletion in long arm of chromosome 13 and in chromosome 20. Some other malignancies associated with structural chromosomal abnormalities, that is translocation between chromosome 12 and 15 lead to formation of congenital fibrosarcoma, secretory breast carcinoma, memory analog secretory carcinoma of salivary glands or mesoblastic nephroma. Translocation between chromosome 12 and 16 is seen in myxoid liposarcoma, and translocations between long arm of chromosome 11 and long arm of chromosome 18 seen in mucosa associated lymphoid tissue lymphomas, and translocations between long arm of chromosome 11 and long arm of chromosome 22 is seen in Ewing sarcoma, whereas translocation between short arm of chromosome X and long arm of chromosome 18 is seen in synovial sarcomas, and translocation between long arm of chromosome 2 and short arm of chromosome 3 is seen in follicular thyroid carcinoma. Now, one of the other application of fluorescent in situ hybridization or cytogenetics is in selection of patients for targeted therapy. Molecularly targeted therapy is a new way of approaching cancer treatment. It involves rational selection of drugs which target specific processes in cancer cells. Targeted therapies are increasingly being used in combination with other targeted therapies or other treatment modalities. Personalized treatment using genomic or proteomic technique is being done by determining most susceptible molecular targets in a specific patient. Examples of targeted therapy are monoclonal antibodies used against certain malignancies like rituximab used in non-Hodgkin's lymphoma, gemtuzumab or ogogamicin used in acute myeloid leukemia, alemtuzumab used in chronic lymphocytic leukemia, ravocirumab used for gastric cancers, pembrolizumab, nivolumab used for melanoma, and necitumumab used in non-small cell lung carcinomas. Applications of fluorescent in situ hybridization in identifying minimal residual disease. Minimal residual disease is a stage in leukemia treatment when patient is in complete remission, that is absence of malignant disease by clinical imaging, standard histologic or cytological means, but small number of malignant cells still persist in the body. These small number of malignant cells can lead to relapse of disease. So minimal residual disease assays guide therapeutic decisions, that is in whom therapy must be continued or intensified to minimize likelihood of clinical relapse. Minimal residual disease predicts outcome of the disease. Level of minimal residual disease at a certain time in treatment is a useful guide to the patient's prognosis. MRD is a powerful and independent prognostic indicator. Coming to summary, each cell in our body contains 23 pairs of chromosomes, that is 22 pairs of autosomes and 1 pair of sex chromosome. Each chromosome contains thousands of genes which direct the body's growth and chemical reactions. Many types of chromosomal abnormalities exist which can be broadly categorized as either numerical or structural. Structural chromosomal abnormalities result from breakage and incorrect rejoining of chromosomal segments. Chromosomal abnormalities can occur as an accident or age of the parent, and certain environmental factors like radiation, infections, drugs, etc. may play a role in the occurrence of the abnormalities. Common structural abnormalities are deletion, inversion, duplication, isochromosome formation, and ring chromosome formation. Balanced chromosomal abnormality is when complete chromosomal set is preserved through rearrangement. Balanced chromosomal abnormalities include inversions and translocations. Unbalanced chromosomes result when there is loss of part of chromosome or there is addition of chromosomal segment. Unbalanced chromosomal abnormalities include deletions, duplications, insertions, ring formations, and isochromosomes. Balanced chromosomal abnormalities may go undetected because they may not result in diseases. A disease can arise as a result of balanced rearrangement if the breaks in the chromosomes occur in a gene and fusion of chromosomal segment results in a hybrid of two genes, producing a new protein product whose function is damaging to the cell. On the other hand, unbalanced structural chromosomal abnormalities may result in spontaneous abortions, diseases, disabilities, various syndromes, mental and physical development delays, etc. Studying chromosomal abnormalities can help preconceptional counseling of the couples and prevent further production of offsprings with congenital anomalies. Other uses of cytogenetics or study of structural chromosomal abnormality are confirmation of diagnosis in leukemias, prediction of prognosis and outcome, selection of patients for targeted therapy, prediction of drug efficacy and toxicity, and monitoring of minimal residual disease. These are my references. Thank you.